Hello Stonites. Today the topic that I'm going to discuss is about ribosomes. Ribosomes is also known as Paladis particles. There are three scientists who plays very important role for the structure of ribosomes and for the studies of ribosomes. One of the scientists is Claude. Okay, he had isolated ribosome. He had isolated means he had taken out the ribosome from the cytoplasm. And he named it as microsome. Okay, it was Claude. For the first time, he had isolated ribosome from the cytoplasm and he named as microsome. The next scientist, known as Robinson and Brown, okay, they had observed the structure of ribosomes in the root cells of Vicia faba. Okay, it's a plant. Then the term ribosomes was given by Pallet. Term ribosome was given by Pallet. Now if you see, where do we find ribosomes? Okay, ribosomes are found both in prokaryotes and eukaryotes. Okay, and both in prokaryotes and eukaryotes. Now, they are also found in other organelles. Okay, they are also one of the organelle, but they are found even in other organelles like mitochondria. So in mitochondria, we have already discussed that there is a ribosome, and that is 70S, right? Mitochondria and plastids. Okay, so these are the two organelles where you find even ribosomes. Usually they are non-membranous. Okay, they don't have any membrane. Okay, like chloroplast and mitochondria, they are membranous structures, right? Double membrane structures. But in ribosomes, they are non-membranous. Now, if you see their attachment, okay, if they are found freely in the cytoplasm, okay, if they are found freely in the cytoplasm, they are said to be free ribosome. Okay, in the cytoplasm, if ribosomes are found, it is said to be free ribosomes. Free ribosomes. In the case of prokaryotes, okay, they are said to be free ribosomes. Now, if you see in eukaryotes, they are found in the endoplasmic. They are attached to the membrane. If it is attached to the membrane, say that endoplasmic reticulum, okay, and nucleus. So if they are attached to any of these membrane, either in the endoplasmic reticulum or to the nucleus, they are said to be bound. Bound ribosomes. In the case of both prokaryotes and eukaryotes, you will find, okay, both prokaryotes and eukaryotes, you will find bound ribosomes. But in prokaryotes, you will find only free ribosomes. Ribosomes has basically two subunits. That is, larger subunits and smaller subunits. Subunits of ribosome. So, large subunits and small subunits. Now if you see large will be attached, okay, attached to the membrane. Is attached to the membrane with the help of a special protein. With the help of a protein, okay, with the help of a protein called ribofourin, okay, ribofourin, ribofourin, okay. So this is a kind of protein, okay, that help for the attachment to the. Just say that if this is your membrane, then there is a protein. This is ribofourin, okay, and the larger subunits will bind, okay. So the binding of the larger subunits to the membrane is held by ribofourin. Smaller subunits are usually free, okay, free in the cytoplasm, okay. And once the larger subunits come to an action, okay, the smaller subunits also comes and bind. The examples, okay, the differentiation between the two types of ribosomes. Now in prokaryotes, it is 70S. Prokaryotes and eukaryotes. So in prokaryotes it is 70S and eukaryotes it is 80S. So what is S? S is said to be sedimentation coefficient. Okay, sedimentation coefficient. It has two subunits, okay? As this is ribosome, 70S and 80S. So it will have large subunits and smaller subunits. In the case of prokaryotes, it is 50S and it is 30S. Okay? Whereas for eukaryotes, for 80S is 60S and then it is 40S. Now, these 50S and 30S, again they have subunits. Okay? Now when I say ribosome is nothing but ribonucleoprotein. Okay, so it means that they are made up of RNA plus protein. So when I say rRNA plus protein, it is nothing but ribosomes. Now in case of prokaryotes, it is around 60 to 65% of rRNA. Okay, and then 35. This is 60 to 65. Then we have 35 to 40% of protein. In case of eukaryotes, it is 45% of rRNA and 55% of protein. Okay, so as I said that ribosomes is composed of rRNA and protein. Okay, it has also some subunits. Okay, so it is 23S, 5S, and then 16S. These two is considered as large subunits. And then 70S is, it will be these two. This is about rRNA. Okay, so these two will be considered as large subunits, and this will be considered as small. Okay. Likewise, for eukaryotes, okay, we have 28S, 5S, okay, and then 5.8S, and then 18S. So these three will be considered as large subunits, and this will be considered as small. So It means that if I say large subunits, it is about, this is 50S, right? It's a part of 50S and this is a part of 30S. Okay, this is what I want to tell you. If it's a, when I say large subunits, it will be a part of 60S, and then smaller subunits means this 18S is a part of 40S. Okay, so these are the basic composition of the prokaryotes. Now we will try to see the structure, the structure of the large subunits and the small subunits. First, I will make larger subunits. Okay, usually there are 3 parts. It's divided into 3 parts. It is said to be E, P, and A. E is said to be exit site. Okay, exit site. P is said to be peptidyl site. Peptidyl site, or we call peptide formation. Peptide formation. And A is said to be acceptor site or amino acyl site. So these ribosomes are responsible in the protein synthesis. Okay, so during protein synthesis we need ribosomes, and this is large subunits of the ribosome. So in A site the amino acid will come. Okay, in A site amino acids, just say A amino acids. Okay, you know that proteins are the, when I say proteins are made up of amino acids. When many amino acids combine or join with one another, we call as protein. So ribosomes, in ribosome in A site, the amino acid will come at the acceptor site, and in P site 2 amino acids will form a bond. And E site it will exit. Okay, we'll discuss this in detail in class 12. Okay, but just try to understand that this ribosome, okay, the ribosome are responsible for the protein synthesis, and it is a larger subunits. Likewise, we have a smaller subunits. The smaller subunits has, this area is said to be mRNA binding site. So once the smaller subunits come, then the larger subunits also come to an action. Okay, in that way the protein synthesis take place. Now let's try to understand the differences, okay, between prokaryotes and eukaryotes. Okay, that is, in prokaryotes the ribosome is 70S, and in eukaryotes it is 80S. Okay. Now, if many ribosomes, even mRNA, is very important. Okay, even mRNA is nothing but messenger RNA. Okay, messenger, or I'll say that this is mature RNA. Okay, messenger RNA or mature RNA. So 5 prime to 3 prime. If many ribosomes, see, if many ribosomes comes and bind to mRNA, if many ribosomes comes and bind to mRNA, we call as polysome. We call polysome. Okay, this question was asked in your medical examinations. Now, one thing you have to know that for the process of translation, when I say translation, means protein synthesis. For the process of translation, when I say translation, means protein synthesis. Okay, we need magnesium ions. So this is about the detailed information about the structure and the functions of the ribosome. The most important function of ribosome is they involve in the synthesis of protein. In the next class, we will talk about plastid and its type. Thank you.